Welcome back. In this video, we are covering enzymes, one of the most important topics in Cambridge IGCSE biology syllabus, one of the topics where students lose the most marks every single year. I'm going to give you the exact words you need to write in your exam, not just the ideas, the precise Cambridge mark scheme language. All right, let's dive in. First, the definition. And this one you must get exactly right. An enzyme is a biological catalyst made of protein that speeds up the rate of chemical reaction in living organisms without being used up. Say that again with me. Biological catalyst made of protein speeds up chemical reaction, not used up. Every single word in that definition matters. If you write enzyme speeds up reactions, you lose marks. You must say biological catalyst and you must say protein. Enzymes are globular proteins. Their three-dimensional shape is absolutely critical because the shape of enzyme determines what it can do. Change the shape, you change the function. We'll come back to that. Now, how does an enzyme actually work? This is called the lock and key hypothesis. The enzyme has a region called the active site. The active site has a specific shape. The substrate, that is the molecule enzyme acts on, has a shape that is complementary to active site. Complementary, that is your keyword. The substrate binds to the active site to form what we call an enzyme-substrate complex. The reaction takes place, the products are released, and the enzyme is unchanged. It can be used again. Now, here is a very common mistake in the exam. Students write the substrate fits into the active site, like a key fits a lock. Do not write that. The examiners want you to write the substrate as a complementary shape to the active site. That is the language that earns marks. Now, let's move on to enzyme specificity. Because each enzyme has a unique shaped active site, each enzyme can only work on one specific substrate. This is called enzyme specificity. Amylase, its active site is complementary to starch only. It breaks down it into maltose. Protease, active site is complementary only to protein. Breaks peptide bonds. Lipase, active site is complementary only to lipids. It breaks fats into fatty acids and glycerol. If the exam asks, why is amylase specific to starch? Your answer is the active site of amylase is complementary in shape to starch only. Other substrates cannot bind to the active site of amylase. Write that exactly like that. That is it for today. So, you can now define enzyme correctly. You can describe the lock and key model using the right language. You can now describe enzyme specificity using the correct terms. In the next video, we are going to look at the factors affecting enzyme activity and how denaturation of enzyme occur. If this video has helped you, subscribe to this channel and thank you for watching.